So thank you, Patrick, to organize this session and to invite me for this talk. So the objective is to share with you our development of nanomedicine. We are developing LivaTag, and the idea is to share this experience with you and to explain how we, along the development, we cross NCL on the road of our development, and how the platform such as NCL could support and help the sponsor in the development of nanomedicine. So a few words regarding the nanomedicine. I think everyone knows that we are designing some nanomedicine in order to improve the drug delivery of the API, to have an efficient targeting. It's this one. More efficient targeting or better intracellular diffusion, or it's a way to overcome drug resistances. Or you could have the opportunity to combine diagnostic for imaging and therapy, and at the end you can protect the API from any degradation in the body. So it's a way to have a large variety of kind of nanoparticles. It could be liposome polymer nanoparticles, dendrimer, micelles, and so plenty kind of nanoparticles with pegylated shell or corner with ligand, or you can imagine what you want in your nanomedicine depending on the target and the objective of this nanomedicine. So that means that it's a major challenge to characterize this product. And we have to consider that if you have plenty of nanomedicine, yours is unique at the end. In the development, you have to consider the regulatory context. And in this field of nanomedicine, we have limited specific nanomedicine-related guidance. But even if you have few guidance, it could help in the development, it could support. But at the end, you need to be creative and to have your own way to develop and characterize your product. So our product is called LivaTag. It's a nanoparticle approach that is ideal solution to address liver cancer. So it's a formulation of doxorubicin. It's a patented nanotechnology platform transdrug. This technology is coming from academic research. The major point of this formulation is to overcome drug resistance, and this product is able to accumulate in the liver and escape to tumor cell resistance mediated by MDR efflux pump. The mechanism of action is adsorption of the nanoparticle at the surface of the cell. We could have the release of doxorubicin very close to the membrane to have a formation of ion pair, and by this way, the doxorubicin is eaten and can enter in the cell, and we can increase the cytotoxicity of the doxorubicin in comparison to the doxorubicin alone. This product is considered as breakthrough innovation in hepatocellular carcinoma. It's a highly resistant tumor with more than 700,000 new cases per year. And the diagnostic of this cancer, it's at the late stage, and you have only one approved product so far, which is sorafenib in advanced HCC. We performed and conducted a phase 2 clinical trial, demonstrated an improvement of the median survival using LivaTag in comparison to the TACE as a comparative arm, this chemoembolization. We have some patents, and Europe and US granted an orphan designation for this product. The product is currently in phase 3. So the objective of this study is to confirm the efficacy, and the primary endpoint of this study is the overall survival. And the study is conducted in Europe, US, and MENA in more than 400 patients. As it's important to have or to address the benefit-risk ratio, we have also a follow-up of the study with a data safety monitoring board in order to be sure that the safety of the product is okay. And we passed successfully 8 DSMB with recommendation to continue the study without any modification. The preliminary result is expected mid of next year. And in parallel, we started a preclinical program to assess any potential other indication or combination of Livatag with any other product. In parallel to the clinical development of this product, we have to focus and to run fast on the pharmaceutical development of the drug products. The objective is to design the drug product in order to meet the patient's needs and requirements and to design the right product with the right performance. And what is important, it's also to design a robust and reproducible manufacturing process. That is very important because usually for this kind of product, you have no non-standard process of manufacturing, and you need to control very specifically this product in order to control your finished product and to be sure of the efficacy and safety for the patient. So it's very important to assure this patient safety and efficacy through the critical quality attributes of the drug product. So you start by the end, you define and you identify the drug product CQA, and you have to control this drug product CQA through a robust and reproducible manufacturing process. So we follow the approach of the quality by design that is described in ICH. So we start with first with the target product profile. So I have just, I don't know if you are familiar with the target product profile, but it's just a summary of the development program. That it's important. It's a living document that you improve and after the different step of the development of your product. And at the end, this target product profile will be the summary of the drug product characteristic of the product. So at the end, you have the claim of your product, and you have to address on all this typical section regarding the indication and any other section regarding the drug product, and it's supported by the clinical study, preclinical study, and information regarding the drug product. So once you define your TPP, you have to define the quality target product profile and identify that it's the most important, the drug product quality attributes. And once you have defined this drug product quality attributes, use all the information you have on your product, all your knowledge. You perform regularly some risk assessments, and the objective is to link the quality attributes of the raw materials, the critical process parameter, to the drug product CQA. And in the way you define the relation between this parameter and the critical product quality attributes, you're able to better control your product. So you can use some design of experiments, and at the end you will be able to have a design space where you're able to clearly control your product. And if you control your drug product CQA, you don't expect any issue in the efficacy and the safety of your product. You're able to design and implement a control strategy. And even if you need for the development, you have to continue in life cycle management. I show you some example of critical quality attributes. If you are injectable product, you have to follow the monograph of injectable product. So sterility, osmolality, everything that you need. All the characteristics in regards to the active pharmaceutical ingredient, assay, impurities, and reconstitution if you have a freeze dry. So you have this information of your product, and at least it's a minimum that you have to follow. And it's not so easy in the case of nanoparticle. And as you have a nanomedicine, you have to add some kind of other critical quality attributes. So this list is not exhaustive. You have to adapt this list in regards to your own product. But what is important, you need to characterize the morphology, at least, of your product, the diameter, the surface change, encapsulation efficiency, and so on. And you need to have a good knowledge understanding of what is your product. In order to manage this characterization, because it's clear that you are P, because you have your drug product EQA, but you need to characterize. And to do this, this work, you have different options, or the most important is maybe to combine all these options. So if you are big pharma or small pharma, you could start with internal department, and if you have expertise and knowledge of your product, you could have an idea and develop a kind of know on your product. And when you move in a clinical study, you have to adapt the quality of your product and to develop some method and to comply with the GMP or GLP requirement to have some QC testing, stability study, and validation according to the regulatory requirement. You could have some, even if it's not GMP, you could work with CRO with the specific expertise that you need for your product. In parallel, you could collaborate with the university because all this literature is coming from the university. So you have a great experience, science in the university, and we have a nice consortium with other companies, with the CEA, with also Amatsi as a CDMO, Nanobiotics as a company as ours, and also a lab at the University of Paris. So it's a way to increase the knowledge of the nanoparticle and to better characterize. And the last but not the least is the platform such as Nano Characterization Laboratory, because you could have some great expertise, the background of a number or plenty of nanomedicine, cutting-edge technology, and you have also the knowledge and some, you know, what could be expected by the authorities and especially the FDA. So it's very useful to have the discussion with the team, scientist team at the EUNCL, and we can expect to have exactly the same kind of relation and discussion with the EUNCL. Why did Onxeo decide to collaborate with the EUNCL? Because we are already in phase 3, and usually you could have the opportunity to have an agreement with the EUNCL if you're at early stage or before the first in man of your product. So the context is we would like to increase the knowledge of nanoparticles and to have a better characterization. And we are in the phase, we use the quality by design, and we are at the same time in the process of industrialization, anticipation of any scaling up. So it's required a very good knowledge and understanding of each step of our manufacturing process. And for that, we need to have, as much as possible, the fully characterization, and we will try to answer to all the questions we have. So our expectation is to have the benefit of the expertise in nanomedicine characterization, scientific support, high level of technology with wide variety of platform, and you have some kind of guarantee that the report that you will have coming from this NCL can be used for your regulatory submission. That is very important. So moving from US to EU, we consider that EUNCL clearly is able to support the pharmaceutical industry, offering a broad expertise, science in the field of nanoparticles, and you have the relation and you can benefit from the US NCL through EU NCL. You could have a package of all the physical chemical characterization, but also immunotox that is very important, or non-clinical in vivo assessments. They will use standardized method. That's also it's important regarding the quality of the data you will collect from this study. So this platform is similar to US, even if it's in a different country in Europe, but it's the charme de l'Europe. And what is also important, this platform will be very close to the European sponsor, even if it's easy to go through Atlantic and meet the team in the US, and it's free of charge. For small company or startup, it's important that you could have the complete characterization of your product free of charge. So better characterization, better understanding, better control, and at the end, it could really serve your product and mitigate the risk in the CMC drug development. Thank you for your attention.
Yes. So thank you very much, Caroline. So now we move to the next presentation.